Thank you, Nick. As you say, we have to come to Valencia halfway around the world to catch up. I want to talk today about our work with DNA vaccines. Many of you will know that DNA vaccines have a poor reputation, but equally they have a number of advantages. They're simple and inexpensive, they're stable, they can be deployed rapidly. Individuals have no pre-existing immunity, unlike many of the viral vectors which have been used in HIV clinical trials. Thank you. They can be used in multi-dose homologous regimens or heterologous prime boost regimens, and they generate cell-mediated and humoral immunity. Although my interests are HIV and hepatitis C, and we think that neutralizing antibody is too difficult, so we are trying to generate cell-mediated immunity. The disadvantages are that they have suboptimal delivery and poor antigen expression. Now, we deliver all our DNA vaccines by intradermal route, so I think we've overcome some of those disadvantages. Unlike ADVAC's and Nick's hypothesis, I think we do need some localized inflammatory responses, certainly for DNA vaccines, and they're not effective in large animals. So I think what I'm going to tell you today, I hope will convince you that we've overcome some of the problems of these two latter points. Initially, we used heat shock 70, HSP70, heat shock protein 70, as an adjuvant. It's been used as an adjuvant previously, but not in this form. We made a number of different DNA vaccines, each of which encoded GAG, driven from the CMV promoter, and different forms of HSP70 driven from the SV40 promoter. We have a V5 tag in the cytoplasmic form, which differentiates between endogenous HSP70, and then we drive secreted HSP70 from the TPA leader, and we have a membranous form that is anchored in the membrane by the transmembrane domain from human transferrin receptor. And these are just the immunoblots, the Western blots, to show you this is a V5 tag from PGAG V5 HSP70. This is the control, and you can see that it's specific. Equally, in the cell culture fluid, we can only detect HSP70, and this time with an anti-HSP70 antibody in the cells which have been transfected with the secreted form. And finally, here's the cell control, and you can see equally the membranous form of HSP70 is beautifully displayed on the cell membrane. Now, we vaccinated mice with each of these different constructs, and we challenged the mice with a virus called EcoHIV. EcoHIV is a chimeric HIV in which the GP120 has been replaced with the envelope proteins from murine leukemia virus, and this EcoHIV replicates and infects mice. So we vaccinated mice, and we're now looking at the viral load in peritoneal exudate cells in the different groups of mice. This is the mock challenge. These are the mice vaccinated with the empty vector that encodes GAG, and GAG plus HSP70. And you can see that there's a statistically significant reduction in the viral load in the mice which were vaccinated with the membranous form of HSP70 plus GAG. Equally over here, you can see that there are statistically significant reductions in the viral load in the mice which received the GAG plus the secreted form of HSP70, and or the combination of the secreted and the membranous form of HSP70. So clearly, HSP70 as a natural adjuvant is having a great effect in these DNA vaccines. Now, we figured that If we were producing one DAMP, that is damage-associated molecular pattern, that is HSP70, if we produced both pathogen-associated molecular patterns and a whole range of DAMPs, and there's a few of them listed here, then we might actually get a double whammy, if you like. We'd induce PAMPs, pathogen-associated molecular patterns, and DAMPs. If we did that, these would be recognized by pattern recognition receptors. We'd go through the process and we'd end up, hopefully, with high levels of adaptive immunity. So in order to try and achieve this outcome, here is a hypothesis that DNA vaccines which encode a suitable viral immunogen and a cytolytic protein will induce necrosis in vaccine-targeted cells, resulting in a release in a range of DAMPs and cross-presentation of the immunogen and an increase in virus-specific immune responses. So essentially, what we're trying to do is to attract circulating dendritic cells to the site of vaccination and induce a degree of inflammation. Essentially, if we can't target the vaccine to dendritic cells, and we haven't learned to do that very well, then we'll attract dendritic cells into the site of vaccination. And the outcome is to actually mimic the effect of live attenuated viral vaccines. Now, live attenuated viral vaccines might be attenuated, but they're still actually cytolytic. So this is what we're trying to do. We've made a number of different DNA vaccines. In this case, we're using luciferase. We do intradermal vaccination into the ear. We drive luciferase from the CMV promoter, as I described before. We have a number of different cytolytic proteins, NSP4 from rotavirus, perforin, and DTA. Now, I don't really have time to describe all the outcomes of these different constructs, but essentially in our hands, the CMV promoter is 10-fold more effective than the SV40 promoter. So essentially here, we get a 10-fold ratio of luciferase to the cytolytic protein. Down here, we get the reverse. We get 10-fold of the cytolytic protein to luciferase. And here we have a polyprotein that is cleaved by the foot and mouth disease 2A protease to produce a molecular ratio of 1:1. Now, we've done a number of studies that actually, in our hands, the perforin gives the best results. And again, I don't have time to go through this in detail, but I'd like to focus down here. These are, I think, if we look here, DTA should be somewhere in here. Oh, here's DTA. DTA kills the cells very quickly, so we get very little luciferase expression. But if we focus now here, this is perforin with luciferase perforin. It takes some time in vivo to kill the perforin-positive cells. But essentially, we're measuring luciferase expression in vivo as a marker of cell death. And essentially, the DTA kills the cells very quickly. The luciferase takes some time to do that. And now, if we switch luciferase for HIV gag, again, we'll look at the viral load in vaccinated mice which have been infected or challenged with the EcoHIV in the peritoneal exudate cells, in spleen, and in peripheral blood. And again, in each of these cases, you can see that the mice which were vaccinated with the gag and the perforin show a statistically significant decrease in the viral load in the peripheral exudate cells, in the spleen cells, and in the peripheral blood. And just as a matter of interest, the CLEC9 positive dendritic cells are statistically increased in the draining lymph nodes, and the CLEC9A cells are the cells which we know now recognize necrotic cells. So again, the vaccine which encodes GAG and the cytolytic protein provides greater protection against challenge with this chimeric virus, EcoHIV. Now, I want to switch for a moment to talk about hepatitis C virus, because it's equally a difficult virus to vaccinate against. In this case, we've made, again, a number of different DNA vaccines. The CMV promoter driving the HCV NS3 protein, NS3 and NS340 driving perforin, just as we did in the last group of experiments with HIV gag. And again, just to show the difference, the CMV promoter driving perforin. Now, in vitro experiments show that CMV that drives perforin, 24 hours and 48 hours, we get quite considerable levels of cell death. Now, if we just simply drive NS3 from the CMV promoter and perforin from the S340 promoter, we still get cell death, but it's attenuated relative to the CMV promoter. But relative to the NS3 alone, it does induce cell death, both at 24 and 48 hours. Now, we've tried quite hard to show the mechanism of cell death, and at the moment, all I can say is that it's not apoptosis. If we look at down here, doxorubicin as a control that induces apoptosis. Now, apoptosis, to our cost, we discovered that annexin 5 was not a specific marker for apoptosis. So we've looked here to look at annexin 5 so that we see 18%. I just want to compare doxorubicin 18% with NS3 and perforin, 28% annexin 5 positive cells. Now, cleavage of cytokeratin 18 by caspase 3, 7, and 9 is a specific marker of apoptosis. If we look at the cleavage event, so this is full-length cytokeratin 18, in doxorubicin-treated cells, which are clearly apoptotic, then this is cleaved to P21. So these cells are clearly apoptotic. The cells which are treated with NS3 and perforin are not. So the cells are dying, but the likelihood is that it's not apoptosis and it's probably necrosis, but there is no specific marker for necrosis. So at the moment, we'll just say it's not apoptosis. So we've done a number of experiments using NS3 and perforin. We've compared them with another specific marker or another cytolytic marker, VSV-G protein, but again, in each case, we see that the NS3 and perforin gives statistically significant increase, in this case in ELISpot for interferon gamma, not quite so much TNF-alpha and interferon IL-2 for CD4, but it does for CD8, again, interferon gamma, TNF-alpha, and IL-2. In each case, NS3 plus perforin shows much higher responses, statistically significant levels of ELISpot positive cells. Now, the crux is, of course, that DNA vaccines don't behave very well or don't work very well in large animals. So we vaccinated white landrace pigs. They were vaccinated with 300 micrograms of DNA encoding NS3 plus or minus perforin 3 times. 7 to 14 days later, we did the PBMCs. Now, we stimulated these again with NS3 specific peptides. Now, the DNA in this case was delivered by a microneedle device that was provided by our collaborators in the US, FluGen. This is the device. It has 6 little needles that are spring-loaded and deliver the vaccine through holes in the needle on the side rather than on the tip. And this is some of the data from FluGen to show that delivery. I've used this little device on myself. It's painless. It delivers the DNA probably around 1 millimeter into the skin. And again, using that device in the pigs, you can see that, where are we? PVAX NS3 plus perforin gives, in the different NS3 peptide pools, gives a faster and higher responses to each of the peptide pools with the NS3 perforin vaccine. So essentially, the conclusions are that the efficacy of DNA vaccines can be improved by co-expression of natural adjuvants, including HSP70, or the induction of necrosis in vaccine-targeted cells. Now, we think this technology is equally applicable to replication defective viral vectors, that is, the adenoviral vectors, which are non-cytolytic and generally don't, although they do induce cell-mediated responses, they're often not particularly good in terms of protection. Now, I hadn't been able to show you these data, but not all cytolytic proteins are equal. Perforin induces non-apoptotic cell death, probably necrosis, and there's a balance between the level and the timing of expression of the immunogen and the cytolytic protein. Essentially, if you kill the cell before there's enough immunogen, you don't get the kind of responses that we want. And for those of you who thought that there was nothing new in DNA vaccines, this patent is just about to go into the national phase. I'd like to acknowledge the people in my own lab who did the work: Branka, Stanley, Judy, Tamsin, Tessa, Jason. Ilya Voskobojnik from the Peter MacCallum Institute in Melbourne provided us with the perforin gene, and Paul Radspinner from FluGen in Wisconsin provided the microneedle device. And I would just let you know that Pamuk Bilsel from FluGen is actually presenting details of their flu virus vaccine tomorrow. So thank you for your attention.
Yep, so we've got some questions. So front row, yep. Can I just start, in fact, ask a safety question? Because we recently did some work with a DNA vaccine that just encoded an aggregation domain and induced apoptotic cell death, and it looked perfectly safe, but I've just heard from our collaborator in Italy that they're actually seeing osteoporosis in the mice, which is caused by, looks like, osteoblastic death. So it looks like the DNA, even if you're injecting it subcutaneously or intramuscularly in the mice, must be going systemically. And therefore, if you're inducing a cytolytic domain, or in this case a trigger for apoptosis, you may get off-target effects that you're not wanting. So any ideas?
Well, in our mice, we generally vaccinate with between 10 and 40 micrograms. So it's a low dose. We've vaccinated literally hundreds of mice by the intradermal route. We've seen no adverse events. The pigs are perfectly healthy. They get 300 micrograms, and we've seen nothing in terms of adverse events or any skin problems in the pigs either. So.
But I mean, I was a bit surprised. But you know, the histology of the bones in these particular mice is very convincing. But of course, the mice remain healthy. It was only a chance observation that you know, they had thinner bones, but they weren't breaking. So it alerts us to sometimes we probably need to look really carefully.
Sure.
It's not just the general health of the animal, but whether there's some off-target effect. So, yeah, fire away.
Yes, there is evidence in the literature that shows that perforin has also a regulatory role. So how can you reconcile, and for CD8 specifically, so how do you reconcile the data that you have? Have you seen any potential regulatory role that perforin might?
You mean as in inducing Treg cells?
Just basically it plays a role regulating even feedback mechanism, the function of CD8 T cells.
Well, no, we haven't, but then we haven't looked particularly. But given that these cells are killed quite soon after vaccination, I wouldn't expect the perforin to hang around terribly long. So we haven't seen any effect. That's all I can say.
Yeah, one last question, just behind.
Have you tried to deliver your cytolytic DNA vaccine to pigs by electroporation, since electroporation is known to dramatically enhance the immune response in large animals like pigs?
Look, we have never used electroporation. I don't think I ever will. I think the Well, the net effect is probably the same, but I think what the electroporation is doing is probably killing cells and inducing some inflammation.
No, electroporation is not killing cells, but it's just leading to a transient increase in the cellular pores. And this is known to dramatically enhance the DNA delivery, antigen expression, and DNA vaccine efficacy.
Yeah, some of us who've done histology would disagree. I agree with Eric. If you do histology where you give electroporation, there's a lot of tissue damage and a lot of cell death. So it's more about that than, I believe, antigen delivery. But I think we'll take that discussion to lunch. So if we could thank all the speakers from this morning, and we'll reconvene here, let's say at 2:10 to give time for lunch. So thanks to all the speakers.